Morning, everybody. Hope everything is fine. Today, I would like to talk about a few basic principles of inheritance and how fruit breeders use it. My introduction is basically I would like to share and explain to you how genetics and dominant genes was discovered and the implications for fruit breeders. In practice, if you cross a nectarine with a peach, you'll find in the offspring only peaches. Now the question is, what happened to the nectarine? It is just gone. Similar, if you cross two peaches and then sometimes you get nectarines in the progeny. How is that possible? You cross two peaches and all of a sudden you get nectarines in the progeny. Now the question is why, and genetics can hopefully explain that. To explain that, you have to go back to the cell of the plant, and especially the nucleus, where all the genetic material is being kept in a plant cell. And further on in the plant cell, there's two processes that take place, meiosis and mitosis. Now, keep in mind the process of meiosis, the daughter cells are genetically different from the parent cells, while meiosis is the genetic the daughter cells is exactly the same as the parents. For plant breeders, the process of meiosis is, of course, much more important. The other thing that you have to keep in mind that with mitosis, the chromosome number is exactly the same. While with meiosis, only half of the chromosome numbers is present in the daughter cells. That's quite important to remember. In the process of the nucleus, they're taking crossover of chromosomes, and that actually helps to distribute the genes from different parents to the new daughter cells, which is going to be sexual. And for plant breeders, the whole process of genetic combination takes place in the process of pollination. In fact, we are basically just doing the job of the bee. We decided which parents we want to use in the cross combination. Gregor Mendel was a scientist or a monk, which actually was also a scientist and interested in breeding. And he worked on garden peas. And he was faced with a similar dilemma. He crossed certain things. And in my explanation, I'm just going to use explanation of roses in terms of purple. So if he crossed two purple flowers with each other, he found that sometimes, well, there appeared a white flower in his progeny, and he couldn't understand what's going on. And otherwise, if he crossed a purple one with a white, all the progeny was just purple, and then he couldn't explain that. But he was a brilliant scientist, and he with careful observations and record keeping and measuring what is going on, he formulate a hypothesis to explain his results. And basically, he used this notification. We talk about phenotype. This is what you see, the purple, and the genotype. So he then used these principles to explain what's going on, where all the offspring is purple. And you use a model, basically, where you get your different genes from your father and from the mother, and then you end up with a progeny like this, where all the progenies are exactly the same, big A and small a. And all the offspring were purple. And then he formulate a concept of the big A gene is dominant over the A. Although in the progeny there is a small a present, the big A overrides the small a, and that's why you ended up with only purple. The same thing if you cross this purple ones, which is heterozygotes for the genes, although it got a white gene, you can't see it in the phenotype purple, and then you get the segregation of 75% purple and 25%. And the model that you again use is again the genes from the father, the genes from the mother, and the combination that takes place during fertilization. And here you can see big A, small a is purple, double big A is purple, All those three are purple and only the white. And then you get this segregation of 70% purple and white and a ratio of 3:1. And keep in mind the A gene is dominant. Although it's present in the progeny, you can't see it in the phenotype of the color purple. Similar, if you cross a purple with a white, then you landed up with a progeny of 50% white and 50% white, the 1:1 ratio. Now, using that model again, we can see that the one parent is heterozygote. It produces a big A and a small a, and the offspring generate into 50% purple and 50% white, a ratio of 1:1. Mendel's laws, he discovered the dominant effect of genes, and he could explain segregation, and single gene inheritance was actually the foundation of his laws. And coming back to our problem of how, if you cross a nectarine to a peach, only the offspring is peaches, we can now explain it using his model, where we can postulate that the peach is homozygotic for the big P, and the nectarines got small n's for recessive genes. And here again in the progeny, if you map it, you can see all the progenies has got PN. P is dominant over the N, so all the offspring is peaches. Similar, if you cross the peaches and you get all of a sudden nectarines, you can postulate that both parents are heterozygous for the peach nectarine gene. They do carry the gene, but you can't see it. Phenotypically, it is a peach because the gene is dominant over the nectarine. And here you get this, again, the combinations take place for the parents, and here we can see the segregation of 3:1. So the genotype that we postulated is actually correct. You get this 75% peach and 25% nectarines. Now, we as breeders now know that white flesh is dominant over yellow flesh. So we can use that. We know how to use the genes. And also the normal shape is dominant over this flat peaches or donut peaches. Similar, red flesh is recessive. And in pears, the red skin color is dominant gene. So the nice thing about this, you can even see in the seedlings from your cross combination, the red leaves, and that's indication is red fruit. So you can just remove the green fruit if you want red fruit. And that brings me to the female tree rearing for crosses in the case of pears, and we do the process of emasculation, only emasculate the open flowers. And then we transfer the pollen with this glass staff onto the flowers. It's only the females, and this is the male part. And keep in mind that the cross combination to get the breeding objectives can be sometimes quite complex. And I'm not going into detail about that, but it can be complex. The whole idea of a cross is to create genetic variation, and we as plant breeders can select the best ones for further use. Just for interest, for the duration of a pair breeding program, year 1, you start with the cross. Year 2, you plant the seedlings out in bags. And year 3 and 8, you plant the seedlings into an orchard. And it takes, in the case of pears, around about 7 to 8 years after you planted the seedlings before they start bearing fruit. Because pears have got a very long juvenile phase. And then you start with your evaluation. You look at the fruit, the progenies of the different combinations. You do measurements. And then you establish these selections, new selections in different new orchards on a bigger scale and doing more results, and also exporting some of the fruit to see what is the market feedback. And then only released when you've got all the information. So the duration of pair breeding program is quite long, 28 years. Finding market gaps for the plant breeders is quite important. Around about 1965, South Africa had only one blush pear with the name of Forel. But then the ARC started breeding, and then we released a variety in 1993 and harvested early. We followed with another one, which was harvested later on. And in 2009, we released Cheeky, which filled the gap between these varieties. And in 2002, we released a new variety called Cape Blush. So now this is important that it's a long harvesting period, so you can supply the overseas market with a blush pears for much longer than was previously possible. The advantages of breeding is you can create new projects, and the other thing is locally adapted varieties, so African conditions. New cultivars can generate some income, filling market gaps, which is important for us, keeping the overseas markets with a constant product for a longer period. And the most important is to keep South African farmers competitive in the overseas markets and worldwide. This is just Cheeky, one of the varieties that the ARC has bred, and it's got a nice logo, and it's got also certain traits. And this is Cape Blush, which was released recently. Now, fruit breeding, I must say, is not easy. Most of the important traits is more than one gene polygenic, so it makes it much more complicated to breed for that. Disease resistance and screening is quite complicated, where you also have to find conditions suitable to select the resistance ones. And the other thing is consumer and producers' preferences keep on changing, and as pearl breeders, we have to keep up with what is the newest trend. The gene pool is very narrow because pears is not available in South Africa. It's overseas where it grows wild, so we have to import those stuff, which make it also difficult. And of course, to release a new variety, it takes a long time. There's so many processes involved, which before a variety can be released. In conclusion, I can clearly say the life of a plant breeder using the principle of inheritance and genetic is really fascinating, and I do hope you agree with me. Thank you very much.
Thank you, Mr. Human. Now, I just want to say it was a brilliant talk, and I hope you can see